microbial fuel cells. Now it's not industrial topic, but now very much attention to produce energy from renewable resources. Next slide. Microbial fuel cells are the fuel cells which use of the oxidized of the organic compounds and produce of currents. What you see the design of microbial fuel cells is contained two chambers of the anodic chamber which operate in anaerobic condition, and cathodic chamber which operate in aerobic condition, and ion selective membrane that we and as a fuel you can use any organic compounds. It's sugar, it's starch. It can sometimes use the waste of human animals, and syngas also now used for operate this device. In anodic and cathodic, it's connect with resistance to conductive wires, and in this example, the fuel is acetic acid. And acetic acid converts to 2 molecules of carbon dioxide, 8 protons, 8 electrons. Electrons pass to cathodes by wires, and the proton pass through ion selective membrane to cathodic chamber, and these combine protons, oxygens, and electrons, and synthesis of the water. This type of device now more than 100 years. The microbial produce electricity. That was now. Next slide. But it's the question how the electrons which produced inside cells pass to the solid anode electrode. And the very long time, only now type of the transfer electrons was the mediated transfer, at which anode chambers contained a small molecule which diffuse inside cells, accept electrons, and transfer to solid anodes. What this example. But it's mediated microbial fuel cells. Of course, this type of the cells have much disadvantage. So if you use as a fuel wastewater, you can additive special molecule. It's such molecule which very it's a shuttle molecule. It's used of dyes. For example, natural red or metal blue. The renaissance of this region beginning on 10 years ago, which scientists found that the bacteria, which named electrogens, can transfer electron directly on anode. It's next slide, please. This bacteria, type bacteria, was named electrogenic bacteria, which transfer direct electrons on electrons. This bacteria have a special very complicated system which contains much of the special proteins, cytochromes, with much polyhem cytochromes. It's cytochromes which contain chrom 4, it's what hem cytochrome, so 10 hems and 1 molecule of cytochrome. So this system is the transfer of electron from cytoplasmic membrane on the outside the cell. This type of the transfer electron is very efficient. So the alambic effectivity of these cells can be 95%. So the microbial fuel cells, it's a very effective transformer energy, but it has some disadvantage. This device, it is very low density of the currents. So power density is very low. But next, please. But during last 10 years, producers much work with improved microbial fuel cells. It's now about 10,000 articles about this device, but all these articles published last 5 to 10 years. But what's very interesting that during this year, power density grew from 10,000 times. 10 years ago, this power density was 1 milliwatt per square meter. Now it's 2 kilowatt per square meter. But interesting, but all this improvement was produced not with microbial cells, but with the device. So it's the new material for electrodes, the special design. For example, it's you see that one chamber electrode. It's anodic chamber, the non-selective membrane, and simple carbon cathode, which exposed for iron. It's very compact, it's very effective, designed for fuel cells. For my surprise, is practically there are no work about reconstruction of microbial cells which produce electrons. I found only one work which from Massachusetts University at company Genomics from laboratory Derek Lowry. It's a very important person in this field, which construct bacteria, buffer sulfurreductans, which more quickly respiration. They amplifies part of the gene which called ATFase, fructolytic part of this enzyme which hydrolyzes ATP. So after expression these genes, the concentration ATP lowly 50% inside cell. After that, cells beginning more quickly for compensation, more quickly respiration and slow growth but these cells now produce more electricity. So maybe it's the limit in not in the produce electrons, but another part, maybe transfer, maybe device. Next, please. It's a different construction of the fuel cells. For example, these cells without membrane for treatment of wastewater. Next, please. So, in our institute, we decided to improve bacterial cells by breeding. First, we decided to construct mutants resistance to antibiotic phosphomycin. Phosphomycin is the important antibiotic which produces streptomyces, it's another bacteria. Indeed, in phosphomycin is the structure analog for phosphoenol pyruvate. Phosphoenol pyruvate is the main, the central, it's the main molecule in the central metabolic way. So we construct the strain, Chavanella anidians emeroanthus, more known bacteria in this field. Next slide. And you have mutants which resistance to 100 microgram per milliliter of this antibiotic. It's a mutant of two types, small and big colonies. Next slide. It's really this mutant weekly rate for utilization of the lactate. Lactate is the standard substrate for this bacteria. That you see is the wild type is mutant. So it's metabolism more quicker. Next time. And this slide, you see the depolymerization of methylene blue. Also, the rate of depolymerization is more than the mutants. It means that these mutants more quickly accept electron for substrate and more quickly take it to acceptor. Of course, it's small examples of the breeding is possible use for improved bacteria. Of course, really in industry, all bacteria which work now in all industry, and produce of mixed technique as the breeding and the genetic engineering. So in vitro, the pure genetic engineering never give the best results enough for industry. But so it's beginning our work is the breeding of this bacteria. And now in our institute we're beginning work with adaptation of salmonella for genetic engineering. Now we have a plasmid, vectors, and transformation, and so on. So it can mutually be used for method for improving this bacteria. And this, of course, microbial fuel cells not produce much energy for common use, but it's very important application. It's the very important application of the first of the treatment of the wastewater, in the food industry and municipal water. It's now already working big pilot plant. And the very interesting application that the sensors which can introduce in the animal and human also. So for work this device you need glucose and oxygen.